again for this invitation to attend your meeting. I've been involved with the Keratoconus group for a long, long time, and I think it's been a very helpful and productive collaboration, and I hope it will continue to be as productive now as it has been in the past. So, the topic I would like to talk with you about is our work on the genetic determinants for keratoconus. So what we've done in the past and what are the next steps in terms of genetic research. So I'm aware of the wide range in the audience here, so I'd just like to, a couple of basic facts about what is keratoconus. Well, it's progressive corneal thinning with distortion of the cornea that can have a very severe effect on the quality of vision. We know that there are both genetic and environmental risk factors. So if we go back 50 years, everybody thought this was entirely the result of eye rubbing. But we now know that there are genetic factors which set the stage for keratoconus, and that environmental factors, which we will talk about, can make it worse. But you have to have the stage set for the environmental factors to work. If you go back 20 years, the figure which was always quoted as to how common it was in the population was 1 in 2,000. We think this is a very large underestimate. So it was 1 in 2,000 was the quoted figure, but now a large population-based study from North Europe put the figure at about 1 in 350. But some particular groups of individuals, surprisingly Pacific Islanders and Maori New Zealanders, have a much higher prevalence of about 1 in 50 with that. So this is partly the result of improvements in the detection of keratoconus. We have much better and more subtle investigations to detect keratoconus, rather than, I think, that keratoconus is becoming more common. It may be becoming more common, as we'll talk about in a moment, but probably a large influence is that we're better at detecting it. That's bad. There's more of it there than we thought there was, but what's good is that there are now, with the advent of corneal cross-linking, which we'll be hearing about this afternoon, there is a treatment which will arrest, in the majority of individuals, further progression of keratoconus if it can be detected early. And so it's not appropriate for everybody, but again we'll be hearing this afternoon the role of early detection and treatment to prevent deterioration of keratoconus. There are 3 variants of keratoconus. So all keratoconus doesn't seem to be the same. There are different variants, and we don't know whether these are different diseases and whether there are different environmental and genetic risk factors which determine this. So the most common are the central keratoconus, where the central part of the cornea becomes thin and protuberant, or a so-called drop cone, where it's below the center of the cornea, towards the periphery, the white part of the cornea. Secondly, pellucid marginal degeneration, where there's thinning right at the edge of the cornea, and also keratoglobus, where there's thinning of the entire cornea. So we don't really know whether all of these are the same or variants, or they should be completely considered differently. Diagnosis is at various stages in the health pathway, and there's a major role for community optometrists in doing this. Is so seeing children or young adults who notice a change in their refraction or an increase in astigmatism, and there are characteristic signs on refraction. And I think in the future this is going to be a target population, is to try and roll out detection to the people who are seeing the vast majority of individuals with visual problems who are community optometrists. So try to roll out techniques and methods for them to be able to detect keratoconus at the very earliest stages. And so there's refraction, but also confirmation are imaging techniques, of which one is the term is Scheimpflug tomography. OCT is another method. So it's a way of imaging the cornea and detecting abnormalities. And the two illustrations there, the one on the left-hand side shows the redness. So it's like, as you would imagine, a topographical map on your Ordnance Survey map where the steepening of the cornea is portrayed as redness or yellow. And so the green is the normal, but in that cornea, there's an area of steepening of the cornea highlighted as red. On the right-hand side is the same individual taken 6 months later, and so the change has become worse, and so that's progression of keratoconus. And that's what monitoring involves, is seeing individuals on multiple occasions and comparing the scans. Again, the prevalence varies. There are ethnic differences, and the writing's very small, I'm sorry, but on that graph, the populations in USA and Denmark are the little bars very low, and then other populations such as India and Iran with dramatically higher numbers for that. And so the inference from that is that in some populations in the world, the genetic susceptibility is much higher. And in the UK, there are differences between white populations and South Asian populations. It's more common in South Asian and black populations than in white populations. In the pathway to determine what's causing keratoconus, this is what we really want to know, is what's causing keratoconus, and if we know what's causing it, we would have a better chance of being able to do something about it. And so the first avenues for research were to look at associated conditions, and on the left-hand side is Down syndrome, and moving across retinitis pigmentosa, Leber's optic atrophy, neurofibromatosis, Ehlers-Danlos syndrome. All of those conditions are associated with an increased risk of developing keratoconus as well with that. And so then looking at individuals, for example, with allergy or neurofibromatosis, and seeing if the genes that cause that condition are in some way linked to causing keratoconus. It's proven, unfortunately, to be a bit of a dead end. It hasn't really given any consistent results. Those are the ones which people talk about. The most common one in the population is the association between keratoconus and myopia. And the numbers there, so there are the axial length, so that's on a little rather small diagram down the bottom. The axial length is the length of the eyeball from front to back, from the front of the cornea to the back of the eye. And the numbers show that in a population, the average axial length is 23.46 compared to an average population of keratoconus where the axial length is 24.47. So a statistically significant increase in the size of the whole eye. So the cornea is predominantly involved, that's what we think of with keratoconus, but there's expansion of the whole eye with that. And so the genes that we think determine keratoconus are potentially involving the whole shape of the eye. Whether the much publicized epidemic of myopia is actually going to make a change in the prevalence of keratoconus, we don't know, but it's a possibility, and people should be perhaps being aware and monitoring for that. The main complication, the visual loss is obviously very severe, but there are complications associated with that, and this acute corneal hydrops, this is the split in the back surface of the cornea. Cornea becomes acutely waterlogged, as on the left-hand image, and that can be occasionally complicated by infection or blood vessels growing into the cornea, which has a major impact on the success and survivability of a corneal transplant if that becomes necessary in the future. And the figures down the bottom demonstrating that there's a major influence on allergy, potentially eye rubbing, on the risk of developing an acute corneal hydrops or acute waterlogging of the cornea. The role of the environment, that this is difficult to work out the relative contribution between the role of the environment and potential genetic factors, because the argument can be turned around for almost all of these factors. And so, number one, there's a high concordance in twins. So identical twins, if one twin has keratoconus, there's a very high risk that the other twin will also have keratoconus. But they've both been brought up in the same household, so they presumably have a shared commonality of the environment with that. But it's good, I think, indication there's a genetic factor. Geographic patterns of prevalence, we talked about that, the differences between different parts of the world, different populations. The role of eye rubbing and sleeping on your face, with your face in a pillow, and atopy. So atopy is a predisposition to allergic conjunctivitis, asthma, dermatitis, those types of things. So that's maybe the same genes causing one or the other, or as we said, that if you have itchy eyes, you tend to rub them. So there must be an interplay between all these factors. And the end story, as we'll be hearing this afternoon, is the story so far, in a nutshell, is that there's some process of slipping of the collagen fibers, one related to the other. And so the collagen fibers there are shown as the black lines. The bonds between the collagen fibers are the little black cross-hatching, and the greeny arrows showing one slipping relative to the other. So when you do a genetic study and you find potential genes, then the next stage is to look to see what those genes are doing, what they're controlling, and do they have any realistic role in the control between the binding between the collagen fibers one to another. So is there a realistic role for the genes that you've highlighted? Biochemistry, different approaches. So we've talked about looking at the environment, we're looking at genetics. Biochemistry, so this is mainly looking at the tears of individuals with keratoconus, comparing with the tears of individuals who don't have keratoconus, and looking if there's any difference, or if an individual has a corneal transplant, taking the corneal tissue to the laboratory and seeing what the cells do when they're cultured, if they're releasing enzymes which are not being released by normal cornea. And so what it shows is that there are increases in cytokines in the tears. So cytokines are proteins which are the sort of agents which act and perform tasks in the tissue. So an increase in cytokines, One in particular is called SOD1, so there's a lot of abbreviations in this talk. So it's superoxide dismutase, so it's a sort of an enzyme, so that's increased. And proteomics is a technique whereby all the proteins are separated in the tissue and compared with all the proteins in tissue that doesn't have keratoconus. You look at the differences, what they're doing. There are distinct differences, but again, the problem is cause and effect. So a cornea from an individual who's having corneal transplantation would normally have scarring, and the presence of scarring obviously is going to change the pattern of enzymes and proteins in the tissue. So it's quite difficult to interpret the role of these. And again, some others, TIMP and collagens are, collagen A, 8, 1 and 2 are potential candidates. And so that's looking at seeing if there's a correlation between the genes and the tissue proteins there. So there's all these sort of potential targets, but none of them has been convincingly demonstrated. The next route from just looking at the tissue is to look at families of individuals. Again, this is a bit small, I'm sorry about that. But keratoconus definitely can have an inherited role. And it's quite common to have one or two members in a family who have keratoconus, but it's uncommon to have several. So it's common to have one or two, but uncommon to have several. To do genetic analysis in terms of families, you need to have several. And so nobody has ever really identified a family big enough to identify a single gene with that. What we have here, and this is with the help from the keratoconus group, is the best we've achieved are these families here, which show between 3 and 5 individuals with that. And we are revisiting this, and so it's pooling this data from multiple reasonably sized families to see if we can identify genes. So we're looking at that. Other people have done this, and so groups throughout the world have looked at the best available families who have multiple members with keratoconus, and they would normally present a result in that we find on chromosome 5, an area which in that family was associated with keratoconus compared to people who don't have keratoconus. And then another group would report their families, and they would say, well, we found this area on chromosome 7. And another group would say, well, we found one on chromosome 14. And the end result is that a dozen different groups have reported a dozen different sites of interest. And so this there on your right-hand side is a sort of diagrammatic representation of all your chromosomes, and in red are the areas that have been reported. And so it's different people have reported areas of interest right across the genome. So the genome is all your genes. And so it's interesting, but it hasn't provided one answer. Why do we think that is? So the human genome is all the genes that you've got, and that's probably 25,000 genes. Not all those genes are actually doing anything in your cornea, they're switched off. And so potentially about 1,000 to 1,200 genes are actually functioning in my cornea or your cornea. And so a lot of them are turned off. So the inheritance is that you will get half of your genes, half of the genes will be contributed by your mother, or will have been contributed by your mother, and half will have been contributed by your father. And so you are a mix of the two. If you consider other corneal inherited changes, such as the picture on the bottom, that's granular corneal dystrophy, That's not keratoconus. It's an inherited autosomal dominant, so that means you have a 50% chance of transmitting it. That's determined by a single change in a single gene. And so that's like you've been dealt a pack of cards in your life, and if you have the ace of spades, you get granular corneal dystrophy. And so that's an autosomal dominant condition. If you are dealt that card, you get it. This is distinct from what we're talking about with, or we think is relevant to keratoconus, is multi-gene disorders. Needing a multi-gene disorder, as you're dealt your pack of cards, and you have to have a combination that gives you 21. And that's 21, that's 2 cards, or more than 1 card, a combination of cards gives you the right score. And you can get that combination through a combination of different cards. And so it's not just one sequence that causes it. And so it's a contribution, and you can achieve the end result in various different ways from that. So looking at multi-gene disorders, which we think is what causes keratoconus, the big breakthrough about 10 years ago with this was a genome-wide association study. And that's done for a lot of these complex diseases. So it's looking at everything. So you don't know where to look, so you look at everything. And you compare everything in your population with keratoconus with everything in a control population who doesn't have keratoconus. And the end result of that is so-called a Manhattan plot. And so that's the colored graph on the right-hand side. It's called a Manhattan plot because it looks a bit like the skyline of Manhattan, which is the illustration below that. And so what the plot represents is one of your chromosomes laid out beginning to end. So all your genetic material laid out in a line from left to right. So number 1 is chromosome 1. Number 2, the blue one next to it, is chromosome 2. Number 3, so on and so forth, right the way along. So that's all the information in a gene shown left to right there. And what you look for then is a peak across that. And you may be able to see on the right-hand side is a peak in the blue. I'm sorry it's a bit small, but there is a peak in that. And that doesn't tell you what the gene is, but it tells you that somewhere along your chromosomes, in this case on chromosome 17, there is something different happening in individuals who have keratoconus compared to individuals who don't. And so it's a clue to tell you where things are and where to look. Unfortunately, although keratoconus is common, it's not as common as a lot of other things, and so it's difficult to recruit the number of individuals that are required. And normally you would require 5 and preferably 10,000 individuals with keratoconus to do this. So right away you're looking at an international collaboration to do this. And that's one approach, is to actually recruit 10,000 patients with keratoconus, or to look at things which are like keratoconus, but are not keratoconus, with the view that this may give you relevant information. And one particular interesting one is corneal thickness. So everybody, when they're born, irrespective of whether they have keratoconus or not, their corneal thickness is different. So my corneal thickness when I was born would be different from yours. And if I have a thin corneal thickness when I was born, in all probability, my mother or father would also have had a thin cornea. Doesn't mean they've got keratoconus, it just means they're born with a thin cornea, or like big feet or little feet, or tall or short. That's just a characteristic. But it's a characteristic which seems to be similar to keratoconus. And so the technical term is an endophenotype. And so looking at corneal thickness, and the other thing you can look at is hysteresis. So hysteresis is bounciness of the cornea, or how flexible it is. And so by looking at the corneal thickness, it's much easier to recruit individuals and look at corneal thickness because everybody's got a corneal thickness, whether it's big or little. And so it's easier to recruit a large study. And then looking at corneal thickness, and with that you'll see there are dozens, if not hundreds, of areas of interest causing this. And then to compare that with smaller numbers of individuals who have keratoconus and see if there's a cross correlation. And this is the same type of study looking at corneal resistance, which is the flexiness of the cornea. And the numbers there, they're the names of the genes which are associated with those peaks. And this is interesting because you won't be able to read them, they're too small, I'm sorry, but one is called ZNF469. That's just the technical name for the gene, and that's, we'll come to that in a moment, that's definitely related to an abnormality of the cornea. And TCF4, which is next to it, is the gene which is associated with Fuchs corneal dystrophy, which is the second most common corneal disease. And so it seems that in some way the cornea can only express a limited number of genes and that they're all contributing in some pattern to the different diseases that we see. So this is brittle cornea. It's a very severe disease. So that's the gene which was highlighted in those studies. And if you look at it in isolation, it causes very severe corneal problems. So demonstrating that we think we're on the right path with this. This is the Moorfields GWAS. So GWAS is Genome-Wide Association Study. This is the seed fund which you provided, that £5,000, which got us on the way with this, and for that we are extremely grateful for that. That led on to recruiting 4,669 cases with keratoconus, which is huge compared to any previous study. So it's an absolute game changer, so to speak, in terms of research into this condition. And I know some of you will have contributed to that study for that, and thank you. So that was 4,600 individuals with keratoconus compared to 116,000 controls. And the result of that, which is now published, demonstrated that there were 36 areas of interest. And so down the bottom is the Manhattan plot, and you see numerous spikes across that scan. And then looking to see what are these spikes doing, what genes in those areas are relevant. And there were 36 spikes, or genomic loci, and they all clustered around functions of the structure of the collagen matrix. You think back to that slide of the slipping collagen. So there was genes which were controlling the collagen matrix of the cornea, and also cell differentiation, i.e. Stem cell pathways in the cornea. So they're making sense. It's sort of a rational story that this is telling. This is the keratoconus GWAS compared, so looking at corneal thickness, that's the bottom one, keratoconus is top, corneal thickness is bottom, and looking if they're all the same, and they're not all the same. And so it demonstrates the limitation of looking at corneal thickness as a proxy for keratoconus. You have to look at keratoconus for this. One of our current PhDs, so it's continuing research, is moving this to the next stage and looking at the actual tissue. So part of our research program is individuals who have corneal transplantation, donating the tissue, and then looking at what the DNA and RNA is doing. So what proteins are being activated, and the two controls at the top. So in the normal cornea, this area, gene 20, it's all quiet, and on the bottom there's a lot of chatter or activity going on. So it's showing that in keratoconus there's a gene in chromosome 20 which is really sending out a lot of signals. So it's showing us where to look for this. So summarizing, there are multiple mechanisms for this, and so our interest is molecular genetics, and on the right-hand side are the environmental factors, and so we're interested in this. I'm taking this forward. Why are we doing this? It's not just to write papers or to get PhDs done. We think it's to have a mature, significant impact in the quality of life of individuals with keratoconus. So the traditional management pathway is detection, spectacles, contact lenses, surgery. On the right-hand side, for any statisticians or mathematicians in the audience, is a so-called ROC curve, R-O-C curve, and it demonstrates that the more tests you have, the better is the chances, the sensitivity of picking up disease. And so if you just looked at refraction, you would pick up a certain percentage. If you looked at refraction and imaging, pentacam, you would pick up more. And so you would move that curve to the left-hand side. And so by doing better tests and potentially in the future having genetic information, we're going to move that curve hopefully all the way to the left, so we'll pick up pretty much everybody at the earliest possible stage. If we pick up individuals at the earliest possible stage with corneal cross-linking, there is then the potential to intervene before there's been significant or any visual loss. So just to halt this whole thing before it causes visual loss. And so there it is. So corneal cross-linking, that's just what it is at the moment, and we're going to hear more about it. I don't think that's how it's going to be forever. I think there's going to be better things coming on stream, but that's just a proof of concept. There is something cheap, safe, which is available to halt the march of early keratoconus in the large majority of individuals. And so down the bottom, detection and early intervention, and we'll be hearing about this. And so we also, another PhD working with us is looking at artificial intelligence to analyze the scans, to pick up changes in the scans or whatever, which are beyond the capacity of the human eye to detect by just looking at it, or to provide analysis for the community optometrists. They take their scan or autorefraction or whatever, and through some central server, the information is analyzed and say, actually, we think this individual has changes suggestive of keratoconus, and then also in the future, incorporating potential genetic markers, so a saliva test or whatever, to say, well, actually, you've got, the scene is set for you to develop keratoconus. So is this going to make a difference? Absolutely. I think there's already evidence that with the introduction of corneal cross-linking, the numbers of individuals requiring corneal transplantation is dropping markedly. That's not all, sorry Ken, it's not all a result of better contact lenses. I think it's partly the result of arresting the progression of keratoconus. So future research, areas to watch, the introduction of artificial intelligence and analysis wheeled out into the community to detect keratoconus. From our personal research, we are getting ready to trying to recruit internationally to do the next round of our GWAS, and so that would be recruiting multiple centres around the world. That involves fundraising, obviously, and patient engagement with that. And also for the other research arm is continuing to look for families with multiple affected members with keratoconus, and multiple, to be useful, is 3 or more affected individuals.